Hello and welcome back to Equity Tutors. Today we are going to be starting a module on gene regulation, and this is the penultimate module in the A-level syllabus, so you are nearly done, so just keep in there. Today we're going to be looking at gene mutations and also gene regulation. So let's get started and let's start talking about gene mutations. So what is a mutation? A mutation is a change in the sequences of the bases within the DNA molecule. And mutation sounds very scary, but actually not all mutations cause a change in the polypeptide and the protein that is coded for by the DNA. Now, the reason for that is because the genetic code is degenerative. And what that means is that one amino acid can be coded for by multiple different triplet codes. To go into that a bit more, if you remember, each amino acid is coded for by 3 bases. So we call that a codon, and that is made up of 3 bases. So, for example, serine can be coded for by TCT, TCC, TCA, TCG. So you can see that multiple different codons can code for the same amino acid. So that's what we mean by degenerative. So there are multiple ways that we can code for that single amino acid. So because of that, if there are changes in the code, it doesn't always result in a change in the actual amino acid that is coded for. And we're going to go into a bit more detail on that in a minute. So the other thing to remember with mutations is that mutations happen all the time and they happen spontaneously. So every day we have mutations arising, but our body is very good at finding those mutations and then getting rid of them, or getting rid of cells that have those mutations. So generally we have lots of mutations occurring all the time spontaneously, but certain things called mutagenic agents can increase the rate of mutation. So these are things like UV light, cigarette smoke, those kind of things can cause an increase in the number of mutations that occur in a cell. Now, some mutagens, which is another way of saying mutagenic agent, can also be carcinogens. So they can be things that cause cancer, but not all mutagens are carcinogens. So just remember that all mutagens are not carcinogens. So let's talk a bit more specifically about DNA-based mutations. As I said, the amino acids are coded for by a triplet codon. So this is 3 base pairs code for 1 amino acid. And the way that the bases are read in the DNA is they're read in groups of 3. There are 3 different types of DNA-based mutations that we're going to talk about today. There are insertions, deletions, and substitutions. First of all, we have insertions. Now, insertions and deletions are both a type of mutation that causes a frame shift, so we call a frame shift mutation. So what happens with an insertion, as the name suggests, is that we put 1 base pair in where there wasn't one before. Now, this might not sound like a big change, but what happens is this then pushes all of the other bases along 1 position. So this will actually change every single codon that was being read, because now the first 3 bases are now actually the 2nd, 3rd, and 4th. So that 4th base is now part of the next codon. So as you can see, this really has a knock-on effect for the entire polypeptide because it affects every single codon in the chain. That might not sound very clear, so do have a look at the diagram because it will make much more sense then how this insertion causes a knock-on effect for the whole sequence. Now, the same thing happens with deletions. So in a deletion, 1 base pair is lost, and again, this causes a shift, so a frame shift in all of the other codons within that polypeptide. So all of those codons get altered, and then that can change the different amino acids that are coded for, so that can then change what happens to the whole polypeptide. And both of these, both insertions and deletions, because they are these type of mutations called frame shift mutations, and they have this big effect on the entire sequence of that gene, these can have really big effects on the polypeptide that is coded for. The other type of DNA-based mutation that I'm going to talk about are substitutions. Now, substitutions are much less impactful on the whole chain. So with a substitution, just 1 base within the DNA sequence is randomly swapped. And because of that, it's only going to affect that single codon. Instead of affecting the entire gene as the insertions and deletions do, because they are frameshift mutations, substitutions are only changing that single codon. So because of this, they often don't have much effect on the whole polypeptide. But there are 3 specific types of substitution that we can have. So the first one is a silent mutation. And this is where one of the base pairs is changed, but it doesn't change what that codon codes for. So as we said before, because the genetic code is degenerative, if something changes to a different base pair, but that codon still codes for the same amino acid, there's going to be no change in the amino acid and in the polypeptide that is coded for by that gene. So for example, if we had a leucine, which was CTT, and one of those, the last T got swapped for a C, so now it's CTC, that actually still codes for leucine. So that would not actually affect what amino acid is being coded for. The other type of substitution mutations that we have are missense and nonsense mutations. These ones are things that do result in a change in the amino acids. So missense mutations, these are often called non-synonymous mutations as well, are mutations in the bases that then do result in a change in the amino acid that's coded for. So for example, if we had a leucine which is coded for by TTG, if that second T was swapped for a C to now be TCG, that would now code for a serine. So it would have changed that one amino acid from a leucine into a serine. Now that can have big effects on the protein, because it might not have much effect, but sometimes it can affect the folding, or it could be in a very important part of the protein, and when this is mutated, it could cause the protein not to function properly anymore. And then the last type of substitution mutation that we have are nonsense mutations. So nonsense mutations are where the amino acid is changed. So again, the codon is changed and the amino acid that is coded for is actually not an amino acid but a stop codon. So a stop codon is a codon that causes the end of the protein. So it causes a halt in the protein synthesis. It signals to the ribosome to stop translating that protein. So if we have a nonsense mutation, then that means that there is now an early stop codon in that sequence. So a stop codon is TAA, is a normal stop codon. There are also different ones. If you have a mutation that causes one of these stop codons to be inserted earlier in the sequence, then you're going to make the protein will be much shorter. It won't be the full length protein that you're meant to have. It will be cut short in the middle or wherever in the sequence that this stop codon appears. So obviously that is going to have a massive effect on the function of that protein, because it now is missing part of the sequence that it needs. So the next thing we're now going to talk about is gene mutations. Now these are mutations that happen more broadly in the entire gene rather than the specific base changes that we've just discussed. So for gene mutations, we can have a couple of different types, again, like we can with the base pairs. So we can have duplications, deletions, inversions, and translocations. So deletion, I think, is pretty clear what that is. That means just one gene out on the chromosome is deleted. So that means that that gene no longer exists on the chromosome. That's the easiest one to understand. We then have inversion. So during this mainly happens during crossing over during meiosis. And this is where a single gene is cut out from the chromosome and it's flipped. So the chromosome still contains the right things, but it's in the wrong direction now. So now that it's been inverted, that means that the original gene can't now be expressed. So inversion will stop the original gene being expressed. It's still present, but it's now in the wrong direction within the chromosome. Similarly, we can have duplications. So a duplication is where two copies of the gene or the section of the chromosome are present on the chromosome. In this situation, whereas before you might have just had one copy of this gene on the chromosome, now it's been duplicated and both copies are now present. So this actually doesn't normally cause any harm, as the original gene can still be properly expressed, and this can actually lead to evolution. So an example of where this duplication can actually be beneficial is for the hemoglobin genes that have different types, so alpha, beta and gamma hemoglobin. And this is because when originally there was one gene to encode for the single hemoglobin type, there was then duplication, and then independently these two genes can undergo different mutations, and then they can result in two different genes that produce two different polypeptides. And as we know with evolution, if one of those can be more beneficial, then that will then be passed to the offspring. So this duplication is actually a very important part of evolution, and as I said before, it doesn't cause the gene to not function because there is a complete gene there present. So the right polypeptide is still expressed, but we just have an extra gene present as well. So the other thing that we're going to talk about is translocations. Now, translocation is where one section of a gene moves to another part. This means that two genes are affected here because we have the effect of the first gene is now not where it should be and is not being expressed properly, and it's also now disrupted the second gene. So basically it's like an inversion, but instead of flipping itself, it moves somewhere else. The gene is cut, and then this bit attaches to a separate gene. So the first gene that has been cut is definitely not functional, and then the gene that it's moved into, that it's translocated to, may now also not be fully functional because it's been disrupted by this insertion. The important thing to know here is if this translocation happens in certain areas of the chromosome, such as in proto-oncogenes or in tumor suppressor genes, it can cause these genes to be disrupted, and the disruption of these genes could cause tumours to form within the body. And we're going to talk a bit more about cancer formation later on, but basically by having this translocation, this affects two genes. Now that's the most important thing to remember. And just to recap, with gene mutations we've got four different types. We've got duplications where the same gene or fragment of the chromosome is repeated twice. We've got deletions where a part is removed. We've got inversions where the sequence or the fragment is flipped within the chromosome, and then we've also got translocations where a fragment is moved to a new chromosome. So now that we've talked about the different types of mutations, let's talk about the effects that they can have. So only a very few number of mutations will significantly alter the polypeptide that is produced. But as I said earlier, the mutations can cause a big change in the tertiary structure of the protein, because even if it's just a couple of amino acids that are different, then we have differences in the way that the protein folds, and this can really affect the function of the protein. And if we have changes in the protein, then this can cause phenotypic changes in an organism. If the proteins are folded differently and not functioning as they should, this can result in changes in how the organism functions and how it presents. Now, an example of this would be sickle cell anemia, where there is a mutation in the beta globulin gene, and this changes the phenotype of the red blood cells, which means that they can't carry as much oxygen as they should. So as you can see, these mutations can have a massive effect on the function of the proteins and therefore the function of the whole organism. So it's really important that our body is able to control and eliminate these mutations wherever possible, and also it's important that we don't put ourselves in danger of developing more mutations than is normal by exposing ourselves to mutagens such as ionizing radiation and also certain chemicals such as nitrogen dioxide. Okay, so now that we've looked at the mutations, let's go on to have a look at how gene expression is regulated. So as you know, the genetic code is translated into amino acids, and these form polypeptides and then form proteins. So if you remember from the previous modules, we call this process transcription and translation. So transcription is where the DNA is transcribed into RNA, and then translation is where this RNA is used to produce the polypeptide using the ribosomes. So what we're going to talk about today is how are the expression of different genes regulated, and we call this transcriptional regulation. So transcription factors control gene expression, and transcription factors are proteins that bind to specific regions of the DNA. So as I just said, from the DNA to become RNA, this is transcription, and this is done by an enzyme called RNA polymerase, because that makes mRNA from the DNA strand. So it reads the DNA strand and it makes mRNA. And the mRNA is then what's read by the ribosome to make the polypeptide. So the RNA polymerase needs to access the DNA and to know when it should start transcribing the DNA sequence into messenger RNA in order for it to be then used by the ribosome to make the polypeptide chain. So as I just mentioned, transcription factors can control this gene expression by regulating the rate of transcription of the DNA. So transcription factors bind to areas of DNA that are called promoters, and these are upstream of the gene that is being transcribed. So the transcription factors are able to bind to the promoter region of a gene, and then the binding of these transcription factors allows the RNA polymerase to bind and start the transcription. So without these transcription factors, the RNA polymerase can't do its job. The important thing to remember here is that the transcription factors bind upstream of the gene that's being transcribed, and they bind to an area called a promoter. Now, transcription factors in general are in the cytoplasm, and then they enter into the nucleus so that they can access the DNA through nuclear pores. And if you remember from last year's modules, you'll remember the structure of the cell and how the nucleus contains nuclear pores. So transcription factors bind to the promoter region of a gene, and this can either lead to an enhancement of the transcription of that gene, or it can also prevent it. So it can either interact with the RNA polymerase to either increase or decrease the rate of transcription. So these transcription factors are really regulating how fast and what is being transcripted from the DNA. So without the transcription factors, there is not going to be any regulation in the gene expression within the organism. So as I said, because the transcription factors are also proteins, around 10% of human genes actually code for transcription factors. And there are very different types of transcription factors. Some of them can enhance the transcription of a gene, and some of them can block it. And different transcription factors allow an organism to really respond to their environment, because as we know, we always have all of the genes within our body, but we don't always want all of them to be expressed. So the transcription factors really help to control the rate and what genes are being transcribed. And they also ensure that the correct genes are being expressed in the correct cells, so that we're not expressing genes for hair in our eyes, for example. So an important thing to remember as well is that DNA is translated from 3' to 5', but it is transcribed in the 5' to 3' direction to produce messenger RNA, so it goes in reverse when the transcription happens. So an important thing to just remember when you're thinking about transcription factors is that transcription factors work by binding to promoters, and they either allow or prevent transcription by either assisting or blocking the RNA polymerase binding to the gene and starting the transcription process. So they're really very linked to RNA polymerase because they kind of regulate how that RNA polymerase binds to the genes and then what that RNA polymerase can do going on from there. So one example that you need to know about for your exams is estrogen. So as you know, estrogen is a hormone, and one of the ways that it has its effects is by regulating gene expression. Now, the way that this happens is that estrogen diffuses through the cell surface membrane into the cytoplasm of the cell, and then it can then diffuse through the nuclear pores into the nucleus. Now, once it's inside the nucleus, it attaches to an ER alpha estrogen receptor. Now, this receptor is normally held within a protein complex, but once the estrogen binds to it, it can change shape and it then becomes free and can leave that protein complex that it was part of. So now that we've got this free-floating ER alpha estrogen receptor, it can diffuse towards the gene and it can bind to a cofactor, which then allows it to bind to the promoter region of the gene and stimulate RNA polymerase and therefore gene transcription. So it can act as a transcription factor for different genes within the DNA. I know that sounds quite confusing, so I'll just go over it again quickly. So estrogen comes from outside the cell and it diffuses through the plasma membrane into the cytoplasm. It then comes through the nuclear envelope, through the nuclear pores, into the nucleus. And once it's there, the estrogen can bind to the ER alpha estrogen receptor, which is part of the protein complex. This causes a conformational change in the ER alpha estrogen receptor, which allows it to dissociate from the protein complex. Once it's left the protein complex, it can then bind to a cofactor. So at this point, the ER alpha estrogen receptor has bound to a cofactor, and together they can then bind to the promoter and act as a transcription factor to encourage the RNA polymerase to bind and begin transcription of the target gene. So I know that's quite a lot to remember, but as you go over that more, it should make a bit more sense. So the really important thing to remember from this is that transcription factors are proteins and they bind to promoter regions upstream of the gene of interest, and they can modulate the expression of that gene. Okay, great. So that is all for today's lesson. So we've covered genetic mutation. So we looked at both changes in the DNA bases. So we had insertions, deletions, and substitutions. And we also looked at gene mutations. So we looked at inversions, duplications, deletions, and translocations within the genes. We then also looked at how transcription is regulated by transcription factors, and we specifically looked at the example of estrogen. So in next week's lesson, we are going to continue on with this module, and we are going to be looking at other ways that genes are regulated, and we will also be looking at how different types of cells have different expression patterns.
Thank you so much for listening. Remember, you can access additional content on our Patreon page by searching for Equity Tutors, where we have a second 30-minute lesson every week, plus monthly bonus content. You can also find us on most social media platforms. We will keep you updated on new content, and you can find us there by searching for Equity Tutors UK. Please like, share, subscribe and comment wherever you are listening, and if you're enjoying, please leave a review. Bye!
Bye!